Thank you for coming, and I'd also like to thank the organizers of the KITP program, Active Solids, and of this workshop for giving me the opportunity to give this talk. I was supposed to attend it in person, but unfortunately I was delayed a couple of days, but I'll be there next week, and I hope to see many of you still there and have the chance to discuss with you in person. So today I'm going to talk about our recent work on trying to understand a little bit better the nature of fluidization and tissue flow in epithelial tissues, particularly, and how that can be guided by topological changes, okay? Before I get onto that topic, I'd like to take a moment to introduce ourselves. I run a condensed matter group at the Department of Physics at the University of Oslo in Norway, and we work on trying to understand non-equilibrium properties and pattern formation in materials that have topological textures. And that range from crystals, liquid crystals, all the way down to superfluids, and more recently I've been quite focused on understanding topological properties in biological active matter. And that would be in particular the topic that I will be discussing with you today. But there is an underlying mathematical similarity across the systems. And if I get the chance to talk to you and you're interested in that, I'll be happy to share with you and talk about the other topics that we're working on. Okay, so tissue fluidization. What is that? First of all, it has been recognized in biological systems by cell biologists that it plays an important role in many underlying biological processes that go from tissue development and morphogenesis to tissue repair, homeostasis, and cancer metastasis. So all of these processes are in themselves highly complex and engage a lot of biochemical and biomechanical processes, but they seem to share in common a feature which has to do with the fact that in these processes, a large scale of cells undergo relative rearrangement. So there is local rearrangement between cells. And that is what is meant by tissue fluidization, right? So the other way is that the cells have transition during these processes from a state of a more solid-like tissue to a more fluid-like tissue to facilitate movement and reshaping or remodeling, okay? So we start from that baseline, but that is an empirical observation, and there are all kinds of experiments that have been performed and have been published. I'm not going to give a review on it, but there is a common understanding that there is such a thing as tissue fluidization. It is important in these processes. However, from a physicist point of view, I think we're still a while in mathematizing that and trying to understand the nature of this tissue fluidization. How does it occur? What is the origin of it? And how does it manifest? And how does it change the rheology of the tissue? Okay, that's work in process, and we've been trying to go a little bit deeper into this topic. So what I'm going to tell you a little bit now is what I've learned from interacting with my cell biologist colleagues, and also from reading this very nice review papers in developmental biology. And it has to do with the two types at the end of the spectrum. There is a spectrum, but at the end of the spectrum, there is two types of tissue flows, okay? One is of mesenchymal origin, and this is all collective flow, okay? And the mesenchymal type is associated to cells being loosely packed, right? As you can see in here, they can exchange neighbors relatively easy, and they are highly migrating, and they have rather irregular shapes, but nevertheless they're elongated. They could be spindle-like shape, but they have a very clear head and tail. And in fact, their way of motion is very much facilitated by the elongation of the cell. And there is this mechanism that has to do with essentially cells crawling on the surface. So they develop this protrusion at the front of the cell. There is a now I can't remember which one is one, or filapodia and lamellopodia, but one of them, there is a front protrusion and there is a rear contraction, and those two together make the cell move in the direction of what is called this rear front polarization. Okay, so there has been recognized that there is such a thing as the rear front polarization, which is related to the elongation of the cell, and that the elongation of the cell dictates the directional motion of the cell. Again, that is the type of mesenchymal collective migration. Okay, there is the other type, on the other hand, which is called the epithelial collective migration. And it occurs in epithelial type of tissues. So what does that mean? Is those kind of tissues that have strong cell-cell adhesion. So the cells have stable neighbors most of the time, they're tightly packed, and that is reflected in the kind of cellular ordering within the epithelial cell, okay? They can also be in a dormant state, yeah? So they, and when they get activated, they have a collective migration that is not necessarily correlated to them elongating and then moving in the direction of their elongation, as it happened with the mesenchyma. It's a bit more subtle, and it's still quite not fully understood. But what it is recognized is that because they form structured, packed sheets of cells, like surfaces, they have what they call a stable apicobasal polarity and bottom-up polarity, which essentially gives the norm of the surface. So the surfaces have a well-defined orientation, a normal to the surface. And in addition, something that reflects their structural arrangement within the tissue is given by the planar cell polarity, which is not the same as the rear front polarity. I've been confused by that quite a while until it became clear to me that those are two different entities. The planar cell polarity is generated by planar cell polarity proteins, and there is the asymmetry of this protein across the cell that reflects somehow the packing of the cells. What it turns out is that this planar cell polarity may also play an important role in the type of collective migration, and that hasn't been explored as much as the other type, the mesenchymal migration. Okay, so I'd like to go a little bit more into it. Now, there is a spectrum of collective migration from the epithelial type all the way from when they are static, and by loosening up the cell-cell adhesion, that facilitates neighbor exchanges in some way, and collective migration all the way to the mesenchymal type, where more or less the cells are moving individually, and then they move in preferentially along the direction of their primary elongation. So to summarize this part where we introduce the concepts, there are two types of polarities that are responsible for directed motion. One is the rear front polarity that has been explored quite a lot in models of in the hydrodynamic models, but also in particle models of cells within the physics community. And we know that it leads to polar flow, flocking, in other words. But then there is the other type, the planar cell polarity, that I think it's a little more subtle. It is a marker of cell organization within the epithelial cell, but it can also generate tissue flow. And what I'm going to show is that the type of flow that it generates is dipolar. Okay, so stay with me for a second and you're going to find out why this is dipolar. Okay, now with these tools in mind, that we have polarities that are important, and there are two types for epithelial, we set up formulating minimal model of interacting cells. Now, the model has been developed by the biophysics group at the Niels Bohr Institute in Denmark a couple of years ago. Didn't get too much attention, but I liked it very much, so I picked it up very much. So I thought there is some meat to it, and I started working with it, and I think I hope that by the end of this talk I can convince you that this model has some substance to it, and it can be used and hopefully can become more spread-wise compared to the mainstream vertex-like models. Okay, so the main idea of this model is that we have point particles. Those are the cells. And these cells are endowed with two types of polarities. Okay, one represents the apical basal polarity, which essentially defines the norm of the tissue surface. And the tissue surface is determined by the alignment of the planar cell polarities of the cells, which are perpendicular or which are sort of in the same plane as the separation vector between the particles. So the separation vector and the planar cell polarity need to be perpendicular to the apical basal polarity to generate this surface. Okay, how do we do that in a minimal model? Well, we introduce a pairwise interaction potential for soft repulsion and also soft attraction. But the attraction right now is anisotropic, and the strength of attraction is modulated by the relative orientation of these polarities through this sort of geometric constraint factors. And this prefactor here, this anisotropic factor, they're phenomenologically implemented, but they make sense, right? We want to have at equilibrium, we want to have when this is maximized. So we have two cells that have maximum attraction between them when their corresponding AB polarities are perpendicular relative to the separation vector. We do the same for the planar cell polarities. We want the planar cell polarities of the two pairwise pairs to be perpendicular to their corresponding AB polarities. And in addition, we also want the relative separation vector to be perpendicular to the AB polarity. Okay, so that dictates this three anisotropic contributions. And once we have this pairwise interaction potential, we evolve the polarities and the particle positions according to an over-damped dynamics, going down the slowest gradient. Of course, you can introduce different relaxation timescales in there, but for simplicity, we have them the same. And there is some noise to let the system not be trapped in metastable states. Okay. Now, what I've done is that I then coarse-grained this model for the case of a flat tissue. So in the case of a flat tissue, the only dynamical variables are the particle motion within the tissue and the dynamics of the planar cell polarities. Both of these are perpendicular to the AB polarity, which is set to be uniform and along the z direction. So we have a flat surface in the xy plane. That simplifies a little bit the math. So I use the Dean-Kawasaki approach to coarse-grain the velocity field and the polarization. And interestingly, what we find out is that the velocity field is determined by the gradient of a source, which for those of you that are familiar with pneumatics and all the hydrodynamic models, we recognize very quickly that this is basically like the pneumatic autoparameter associated with the polar field. Okay, but this is the polarity corresponding from coarse-graining the PCP polarity, so not the usual type. Okay, but it's still a symmetric traceless rank 2 tensor that gives rise to dipolar flow. But the nice thing about this one, and so there are some models, hydrodynamic models that sort of more or less postulate this kind of dipolar flows in polar systems, but they are quite postulated by symmetry argument, like at hydrodynamic level. So the nice thing about this exercise is that we have a microscopic model, so we sort of trace down to the cell-cell interaction what is the source of this dipolar flow. And that has to do with anisotropic cell-cell adhesions due to misalignment of this PCP polarity relative to each other and to the separation vector. So that's an interesting insight. Does it work? How do we test this? Well, we can set up the planar cell polarity with the topological defect. That is a polarization field. So it will have as its elementary defects the topological defect with charge +1 and -1. Now the +1 defect is interesting because the structure of the polarization depends also on the far field orientation of the polarization. I'm not going to go too much into the details, but there is a subtlety that we can discuss afterwards. But there are two flavors of +1 defects. There is the vortex, there's the spiral, and there's the aster, right? And the -1 defect has this fourfold symmetry. And what I'm plotting here is the vector of the polarization in the px, py space, so in the autoparameter space, around a loop circumventing the defect. So you see that the polarities are rotating. Okay. Mathematically, we can write the polarization outside of the defect core as a unit vector with this phase that is multivalued at the location of the defect plus the far-field uniform phase. Okay. Now we plug this polarization into the formula of the velocity field induced by the dipolar flows. And we find out that the velocity decays as 1/r in the far field and has a false symmetry that is interestingly misaligned with respect to the false symmetry of the PCP polarization. Now, if you look at the positive defects, you see they are basically also positive defects, yeah? But they are rotated relative to the orientation of the polarization. Only for an aster, we have the PCP polarity as a marker of the directional flow. For the other ones, the orientation of the polarization is not necessarily aligned with the direction of the collective motion. For the negative defect, we have this eightfold symmetry that develops. Now, we've also done discrete simulations of the actual particle model in a disk, and we see some boundary effects that we quantified, and we see pretty much exactly the same profile. For the negative defect, we also allow the surface to relax, and we see that it relaxes to this pillow shape, essentially a shape that fulfills the Euler characteristic for the confinement, for the shape of the confinement. But that tells us that the coarse graining procedure is correct, and it captures the dynamics. Now, as I mentioned, there is another model, hydrodynamic model, that has been proposed. There's actually a couple of hydrodynamic models for polar systems. There's one that I've recently been involved with, together with Amin Dusmahadi from Niels Bohr Institute. And what we did in that case is that we also have a dipolar flow that is postulated by symmetry principles. But now the velocity field is subject also to frictional damping and viscosity. And there is also the incompressibility. So the math gets a bit more complicated because the velocity field is the convolution of the active stress. But at the end of the day, the interesting thing is that the negative defect keeps its false symmetry, and actually the far field also decays as 1/r, but it's modulated by the hydrodynamic screening in the far field. So that's the same. The positive one is kind of interesting because all these different kinds of structures in the polarity collapse into only vortical flow. And another interesting thing is that this vortical flow has a flow reversal in the limit where the dissipation is dominated by viscosity. So screening, and we know this also from the nematic, that incompressibility is an important constraint, and also hydrodynamic screening is also an important constraint. So in other words, this flow reversal occurs only in the limit where the viscosity dominates. So if you have friction, we only see if you have friction and incompressible flow, we only see vortical flow, but no flow reversal. And if we have friction dominated and no incompressibility constraint, just like we have it in the particle model, then we have a flow profile that sort of mimics the structure of the PCP polarity. Okay, that's kind of an interesting result. Now I have a couple 10 minutes left, and I'd like to tell you a little bit more about the effect of geometry. The nice thing about this model is that we can simulate it in 3 dimensions. So we can relax the condition of a flat surface, which means a uniform and collinear AB polarity. So we can study the effect, the coupling between topological defects and curvature. And this is a snapshot of simulations where we seed an aster in the PCP polarity, and we see that the surface remains flat, so it's stable to this topological constraint. But if we see the vortex, that leads to an incoming flow, and the incoming flow leads to stress accumulated at the site of the vortex, which leads to growth in the third direction, and we see the formation of this mount. That's interesting. But there's two kinds of things that appear in the discrete model. We have the choice of allowing for neighbor exchanges or not, by basically freezing the connectivity matrix between the cells, or having some condition that when another cell comes close enough, we say that there is a pair exchange. So there's a geometric positional condition to allow for that. Now, if you freeze the connectivity matrix, then interestingly, and we pin this vortex in the PCP polarity, then we are in the solid-like deformation behavior, and we see a relaxation towards a conical shape. And then everything stagnates. There's no more flow. However, if you allow for neighbor exchanges, so if another cell is coming closer than a pair, then we exchange the neighbor to the one that is closest to you, and a new pair is formed. If we do that, then can I simulate this? Yes. Then what we see that it starts forming a cone, but then the cone runs off and it creates a small hole. And around that hole, there is a lot of neighbor exchanges that are happening. And also you see very strong localization of the neighbor exchanges, right? With the mesenchymal tissues, the fluidization is much more spread out, so in some way less localized. But for epithelial tissues, it's highly localized. It almost forms like shear zones, like for people from, fracture mechanic and granular media. So it's very localized. So that was very interesting. So the map that you see here is the number of T1 the number of neighbor exchanges. It's like a density field of neighbor exchanges. So if it's 0, it means that these cells are in the solid state. They deform with no neighbor exchanges. And if it becomes colored, then it means that it has undergone neighbor exchanges. And you see that most of the neighbor exchanges are happening at the tip of this tube. So then we wrote this paper. We said that it's a spontaneous tube formation. You don't need anything other than this allowing the tissue to fluidize, and it will happen spontaneously locally, and it's induced by topological defects. The nice thing about this model is that we can take it again to 3D, and we can go beyond simple layers. We can study multiple layers, and we can also study gastruloids. We have a conglomerate of cells that are polarized, and we can look at their elongation, so we can look at organ development. That is a very ambitious thing, but we go into that direction. Okay, I'm not going to say more about that right now. So for the last 5 minutes, I think that I have, I want to tell you a little bit more how tissue fluidization occurs in different kinds of models. And another model that we've been experimenting with is this multi-phase field model. Essentially, each cell has a deformable shape. It has a shape, no more polarities as primary objects, but it has a shape that can be deformed. And through shape-shape contact, there are adhesion and repulsions. And we have a tightly packed system like that. This is the equation of the phase field for each cell. So they get advected with their self-propelled velocity, which has a constant magnitude and an angle that satisfies the V-check-like model. So it has alignment. And this is the relaxational term that is given by this molecular type of forces determined by a free energy that has pairwise interactions. It has a Angheluta type of surface energy, and it has repulsions. Okay, so what you see here that in this model, we don't need to put in the geometrical constraint as you would do in a point-wise model, a particle model. Here it happens when you have 4 cells that are adjacent to each other, and then two are in contact and two are not in contact. And then through what's called a T1 transition, these two vertices here, they annihilate and then they recreate in an orthogonal direction. So we call this a T1 transition. It is a topological rearrangement because you're destroying vertices and recreating them. And it is considered as being a primary motor for remodeling the cell-cell connectivity. Okay, so then that is the mechanism by which cells are exchanging neighbors through these T1 transitions. So then we are interested in what is the flow profile induced by such a T1 event. Now this is like a shear transformation zone. It has like in granular matter, so it comes for a little bit and then it disappears. But for the time that it stays, then we collect all of the T1 transition. We found an algorithm to reorient them based on the axis around which the vortices disappear and then they nucleate. So we align those principal axes. And we can then average over the cell flow around those. And we find that it has this quadruple pattern. We can also define a polarization, but it's not the same type of polarization. This is more like the rear front polarization because it has to do with looking at the direction of the principal elongation axis. And we see that it mimics the cell motion. So there is a little bit of polarization, but like in all of this multifacial model, the degree of elongation is within the noise, basically. I mean, there is always like a largest eigenvalue, but the difference between the largest eigenvalue and the next one is very small, so the degree of elongation is not persistent. Anyway, the other thing that we can do is to look at how the T1 transition enable the dispersion of cells. So we measure the mean square displacement and the relative displacement as a function of the cell propulsion speed. Yeah, so you see that for the mean square displacement, which is the statistics of single particles, they go they have a slope that is higher than 1, so they are super diffusive. And then they approach the ballistic limit for very large activity, which kind of makes sense, right? Because it's almost like a free path. But it's interesting that that happens in a very crowded environment, okay? So they don't quite bump into each other. They have a way of making room around each other. So the super diffusive behavior, it wouldn't happen if you didn't allow for T1 transitions, for relative migrations to happen, okay? So the other interesting thing is that we try to collapse the mean square displacement and the pair separation as a function of the cumulative number of T1 transition at the given time. And we don't see a good collapse for the single particle, but we see a really stunning collapse for the pair separation distance. And we've done a lot of parameter changes, the activity, the surface tension, all kinds of, there's lots of parameters in this model. And consistently, we saw this robust scaling with the slope of 1, which means that the rate of dispersion is really controlled by the rate of T1 transition. In this model, this is what the fluidization is. So we thought that that's also an interesting result. So I think my time, I have 2 more minutes, but I'm going to hopefully have more conversations with you. So to summarize, I want to highlight the main points. And one is that, planar cell polarity is also very important, maybe even more important than the rear front polarity in epithelial tissues. And within this idealized minimal model of interacting polarized cells, we found out that the planar cell polarity is a source of dipolar flow, and it has this particular pattern. I've also shown you that if we allow for 3D tissues, when the planar cell polarity is imprinted with the pin vortex at its site, that generates spontaneous dipolar flow. And the flow is more like a relaxational type if we don't allow for remodeling of the cell-cell connectivity. In other words, no neighbor exchanges. But interestingly, it forms tubes if we do allow for neighbor exchanges, so fluidization. And the formation of tubes occurs when we have a very localized fluidization at the tip of the tube, and also a type of fracture-like pattern within the tissue, which we don't quite understand. This is very recent work, so we're still digging into it. We've also, I didn't show you to it, but we are comparing between these two models, and we found out that for this discrete particle model, the type of neighbor exchanges is basically like a T1 transition. It's very much the same. It didn't have to, because we only take into account the third neighbor that is coming and destroying the pair. But if we have the condition for having a T1 transition, that is also implicitly satisfied. So that is interesting, which means that T1 transitions are primarily in the main sources of neighbor exchanges, and that they control the rate of relative migration, hence the rate of fluidization. So with that, I'd like to thank my collaborators at the University of Oslo, my postdoc and my current PhD student, my former student, and my cell biologist colleague, from whom I've learned quite a bit of biology in the last few months, and also to Alex Voigt from Dresden. He's been very active on the multi-phase field modeling project. And of course, thank you for your attention, and I look forward to hearing your questions. Thank you.
Thank you. All right, questions for Luiza? I see David already has one.
So thank you for the lovely talk. I was particularly intrigued to see the spontaneous formation of cones, which I will hopefully talk about on Thursday. I had another question, which is in these migrations, these mesenchymal and epithelial migrations, what is the effect of a gradient in some sort of chemoattractant? Do the defects respond to that kind of trigger for directionality? You just sort of impose the directionality at some points in your talk, but how does that react back? I didn't see another equation for the nutrients or the whatever is driving the migration.
Yeah, very good question. Yes. So in the close graining thing here, I've only mentioned about the effect of velocity, the effect of planar cell polarity onto the velocity. But of course, there is the feedback. So there is also an evolution equation for the polarization, which I've just derived, but I'm not yet comfortable presenting it because I need to test it with the discrete particle model and make sure that everything is okay. But we have there is a coupling between them. It's all, I mean, you could kind of see it in the discrete particle model that the polarities act onto particle motion, and the particle motion act on the polarity through this anisotropic term. But it's very interesting the way they come here. So what I find is something that looks very similar. I mean, there is a molecular field, and then there is all kinds of alignment terms that emerge. And I'm still struggling a little bit with finding the alignment terms. They don't, I mean, part of it agrees with very much with the hydrodynamic model of polar system developed by Christina and the others. There is a paper from a couple of years ago where they also use a close graining of a generic polar model. And the molecular part agrees. I mean, yeah, the short answer is that there is a coupling, yeah, but we're still trying to understand it.
I look forward to talking next week. Thank you.
Yeah, thank you.
Great, the next question here.
Hi, thank you for your talk. So in the multi-phase field model, in the two-dimensional case, you showed us the exchange of neighbors, slide 18, I think, or something like that.
Mm-hmm.
Perfect. So here, when the neighbors are exchanged, inevitably the number of sides of these polygons also changes, which has been let's say, related to the creation or annihilation of topological defects. Have you, and it has been shown actually that the flows generated by these defect quadrupoles, let's say, generate a flow that is very, very, like it's exactly the same like the one you showed in the next slide. Have you taken into account the effects of those topological defects in this case? Because before you showed us the vortex-like defect and its effects.
Yeah, I think you're referring to a recent work by Luca Giomi
Yeah, but
and the others where they have a different type of model. And what they show is that, you know, at this scale, one could also, well, we haven't looked into it, but we haven't been in contact with them. So we are aware of the fact that they've also predicted something like that from unbinding of hexatic defects. So one could,
right.
and I think Axel is working on trying to describe the hexatic order in such a system. And what they found out is that when there is a T1 transition, there seems to be an unbinding of plus minus hexatic defects.
Right. I'm aware. I was just wondering whether or not you took
Right.
into account. Thank you.
No, that I haven't. No, that
Okay.
I haven't.
That's
No. I mean, I think this two works came more or less in parallel. We sort of found each other on archive afterwards, and yeah.
Okay. Thank you.
Great. Then next talk, Jose, and then we go to Silke.
Hi, thank you so much for your talk. And I find very appealing indeed this model where you look at planar cell and apical basal polarities and how that helps organize tissue structure. To me, that's very appealing. And I was hoping you could dive a little bit more into the microscopic mechanisms of how cells enforce these polarity vectors. Because I imagine there's a lot of interplay between the polarity, the polarity that's established together with contractility and motion, and potentially also how to connect these microscopic mechanisms to all these coupling constants that you need for your coarse grain model.
So if I understand correctly, the question is if there is more more insights as to how these coupling terms are formulated.
Right, and yeah, just what's the cell doing to enforce, like say you were to perturb cells so that their PCP or AB vectors were pointing in different directions, what would the cells do to correct that? And how would those mechanisms help you understand what these lambdas are?
That's a very good question, and I think that's where the model is in some way phenomenological, because we could put anything that we want. So to my understanding, this is a way of controlling the cell-cell adhesion depending on the local orientation of the PCP polarities. Yeah. So if you have two PCP polarities that are aligned in the same direction, those two cells would tend to form bonds that are stable. But if they're misaligned, it's more likely that they will exchange partners. That's one way to look at it. Now, what are the actual the even more microscopic mechanism for that? I think I'm not there yet. I took this model as this is the level of understanding, and what can we do with it? The principles, I mean, the principle of developing this model is quite simplistic, and it comes from this idea that you have this AB polarity that determines the surface orientation. So it must be perpendicular to all the other degrees of freedom inside the plane. So you have the relative separation, you have the other polarity. And the other important thing is that for the PCP polarity, together with the separation vector, to form a surface, they cannot be collinear, right? They have to be at an angle, because otherwise it's degenerate. So you need two vectors at an angle to form a plane. So it's mathematically very simple in that way, but to put more biology into it, I think I need to talk more with my colleagues.
Well, hopefully we can chat some more later.
Absolutely. If you have some more insights into how to
Great, then Silke.
interpret it, yeah, that would be
Yes,
great.
hello. I'm also very intrigued by this minimal particle-based model of migration. So if I understand it correctly, it's more like a system with two sets of orthogonal dipoles having like an first-order terms of the expansion of a potential. So I was wondering, is from your equations of motion, it doesn't look like this is active. Is this is overdamped, right? Does it stop moving, or is it within migrate?
Very, very interesting question, and that actually points to something that I'm also a little bit. Okay, so it's not active in the sense that what is active actually? In the sense that there is a self-propulsion, because that requires a polarity, but that would be the rear front polarity that has its own evolution, and that would feed into. So we don't have that degree of freedom. We don't have the rear front polarity that it's
But
using to
even if you were
understand.
saying it's not self-propelled, let's say it's an active stress, you would expect some sort of internal equation where it reacts to the other cells or to its environment.
Yeah, but that is the interesting thing. When you cause grain, you get this dipolar flow, which in hydrodynamic models, the coefficient in front of the dipolar force is activity. So that would be an activity. So that means that there is an activity also intrinsically generated by the planar cell polarity in the way that the rear front polarity would generate it. Yeah?
But is there some sort of feedback in your model then? I didn't understand this, or is this just a set potential?
It comes precisely from these cross terms, really. It comes from the fact that this PCP polarity between adjacent cells, between neighboring cells, are not parallel, are like at an angle with each other. That generates effectively a propulsion. The cells do not want to be next to each other if their PCP polarity do not point in the same direction. That is a form of activity.
But the polarities change as a function of time?
Yes. Yeah, they all evolve.
Okay, but I look forward to talking more to you.
Yeah, me too. You will be there next week, hopefully.
Great. Then I also have a question, which is a follow-up to Silke's. So could you tell us how gamma, this coefficient which corresponds to the active stress, relates to the microscopic parameters of the model, to the lambdas, let's say?
They are determined by lambda 3 and lambda 2, essentially. But also when you cause grain, you essentially integrate. There will be like an integration kernel that integrates over this soft interaction potential, this part here that depends on the separation distance. So it gets a prefactor, because in this numerical simulation, we use the exponential, but actually the connectivity matrix is nearest neighbors. But when you cause grain, you just have a generic. So I did the cause graining for a generic short-range repulsion and long-range attraction. So then you get the integration of these terms that modulates the lambda 2 and the lambda 2 and the lambda 3. But they are, so up to an integral prefactor that converges, they're determined by lambda 2 and lambda 3.
I see. Thanks.
Yeah.
Any other question for Luiza?
Yeah, I mean, this is just a reflection of my ignorance. I missed exactly where the breaking of detail balance or something like it went in that allows a system in which you have defined interactions apparently via a pair potential to give rise to a velocity proportion to div of the trace remove part of PP. So there's something like, gamma is something like an activity parameter, but if you could say exactly where you broke time reversal, it would be nice. But others may have got this. I just missed it.
Now that is, I don't know if I can answer that question yet, because these are very fresh calculations, and I was wondering if I should include the slides, but I do want to, because I want to get your feedback. So I know the math is correct, it's checked, but there are some subtleties, and one is exactly where do you drop it. And I mean, the only thing that I can see here is that we have a very anisotropic attraction.
No, but that can't do it by itself.
No, I think it's when the neighbor exchanges happen, when you have the T1 transitions, you jump from a state where they are in the preferred alignment and orientation, right? So they are low potential. Once you exchange neighbors, the new neighbors are in the high potential state again. So you're injecting energy in the late neighbor exchanges. And so that's also how it can keep flowing. If you were to forbid neighbor exchanges, it would just freeze when the energy with the current topology is minimized. But then you're changing the topology, and that's what.
Yeah, that's a good point. That's a very good point. Yeah, so but I don't know where the neighbor exchanges are coming in the coarse-graining. I don't have a control over that. Because I do agree that
Is probably
extension.
just looking at the instantaneous velocity. It's not looking at a long-term dynamics.
That's
It's
true.
taking a
That's
current
true.
state and looking how it will evolve into the future for a little bit, but then it doesn't take into account that it would actually go to the minimum and then sit there unless you change the topology again.
True. True. Yeah.
Great, then thanks Fritjof for the comment, and I think it's time to move on, so let's thank Luiza again.
Thank you.